Good morning, everyone. It's so great to see you all virtually. Hopefully, I'm going to pretend you're all in the audience and that I'm directly speaking to, because I do wish I was there, and I wish we can all hug each other and celebrate some of the greatest things that have happened for the Angelman community in 2020, despite the horror of what has occurred with COVID-19. But I'm really excited to share with you today some of the updates that we have in the research stage of Angelman syndrome. And I really would like to start off by thanking Dr. Meeker. Doug, that was a wonderful talk and really inspiring. And we're really grateful that you were able to share some of your insights with us today. And hopefully we can see going forward a path that will grow similar to how you described for Angelman syndrome. So today I'm going to talk about Angelman syndrome therapeutics. And I've given similar talks before, and I do find that this is really empowering to parents. And the goal of this talk is to be able to share with you where the landscape is in Angelman and the basics behind Angelman syndrome genetics. If you understand the basics behind genetics, you can understand the basics behind therapeutic strategy. And if you can understand this therapeutic strategy, then you can really put your head around the different pharmaceutical companies and their platforms and what is being proposed for us to be able to consider therapeutic treatments for Angelman syndrome. So I really want to just give you a good overview of Angelman syndrome and where we are today, and hopefully where we're going in the future. So for those of you who don't know, my name is Allyson Berent. I am the Chief Science Officer for the Foundation for Angelman Syndrome Therapeutics. I'm also a veterinarian. I do a lot of clinical research, and I help to translate medical device development in animals in order to be able to use that for therapeutic translation, potentially in pediatrics. So I have a good sense of clinical trial design, clinical trial development, and ultimately clinical research. In addition, I am also the Chief Operating Officer of Genetics Biotherapeutics. I'm going to start off the talk today by showing you a really gorgeous video that was produced by David Belinsky. You saw a really beautiful video he made last year as well, and we commissioned him to make another one. This one is really focusing on the basics and the genetics behind antisense oligonucleotides. The focus of my talk is not on antisense oligonucleotides, but because we do have two therapeutic strategies using this technology in the clinic, we thought it would be really important for the parents to understand how these therapeutics work. And so I'm going to start with this beautiful video, and then I'm going to go on to talk about the genetics of Angelman syndrome.
Angelman syndrome is caused by the loss of function of a single gene, UBE3A, on chromosome 15, which is vital to how the brain controls speech, movement, and learning. The UBE3A gene codes for UBE3A protein, which tags other proteins in our cells for recycling in a process called ubiquitination. In our central nervous system, only our mother's copy of the UBE3A gene is expressed or active. The copy inherited from our father is silenced by a mechanism called the ANTISENSE transcript, where neurons manufacture a strand of long, non-coding RNA called the UBE3A ATS. That is positioned on top of the functional paternal UBE3A gene, which results in it being silenced. The UBE3A antisense transcript prematurely terminates the reading of the paternal copy of the UBE3A gene, resulting in a failure of paternal UBE3A protein production. Research funded by FAST is investigating a promising strategy that uses a technology called an antisense oligonucleotide, or ASO, a mixture of DNA and RNA designed to bind the RNA of the UBE3A antisense transcript in an exact location to unsilence the paternal copy of the UBE3A gene. The ASO's sequence of RNA and DNA bases is arranged in a complementary order to the RNA, inhibiting the imprint. The ASO preparation is administered through a lumbar puncture into the spinal fluid. The drug travels through cerebrospinal fluid and flows into fluid-filled structures within the brain and spinal cord to affect the neurons of the central nervous system. Through a process called endocytosis, the ASOs enter neurons of the brain and spinal cord, and then move to the cell nucleus to engage the chromatin, large structures composed of tightly wound and packaged DNA. Because the ASO sequence is complementary to the UBE3A antisense transcript, it can bind to that precise segment and form two complementary strands, which results in cleavage through the RNase degradation. Naturally occurring cellular enzymes aid in the ASO's bonding to and degrading the RNA molecule that is blocking gene transcription, inhibiting the imprint and activating the paternal copy of the gene. Released from blockage, the now bioavailable paternal UBE3A gene is available to be transcribed into mRNA, which then threads into protein manufacturing ribosomes. Ribosomes build functional paternal copies of the UBE3A protein to replace the non-functional or missing maternal supply, and recovery of proper neuronal function can hopefully begin. These research strategies and others have demonstrated positive outcomes in animal models of Angelman syndrome. Two of these strategies are now in human clinical trials in 2020, as we march ever closer to curing Angelman syndrome.
So I thought that was a really, really great video, and I thought it would be great to share just so you can understand some of the strategies we're going to talk about today. So let's talk about the introduction of Angelman syndrome. What is Angelman syndrome? Well, as we all know, humans have 46 chromosomes, so there's 23 pairs. We get one from mom and one from dad. And all of our genes are made out of the material that's in those chromosomes. And Angelman syndrome is caused by a reduction in the expression of a single gene. And that single gene is on chromosome 15, as you can see here. And we get one copy here from our mother and one copy here from our father. This event, which we'll talk about, occurs in 1 in about 20,000 births, so anywhere from 12 to 20,000. And it's typically random, it's typically non-inherited. And it is a de novo either deletion or mutation or some other more unique genotype that occurs, resulting in the loss of function of that gene, UBE3A. All of these chromosomes are made up of different genes, and all of these genes essentially make proteins, and those proteins are what is required for our body to function normally. And so by our children missing the UBE3A gene, that results in a tremendous amount of dysfunction. So Angelman syndrome, like I said, has one copy of the 15th chromosome from mom and one copy from dad. And let's make this a little bit more schematic. So when we look at the chromosome, we can see that there are many, many, many different genes on a chromosome. And this is just one example. And you can see there's a maternal copy of the UBE3A gene here and a paternal copy of the UBE3A gene here. But something very unique happens in neurons. In neurons, the paternal copy of the gene is silenced. And that silencing happens through something called imprinting by something called an antisense transcript, or the UBE3A antisense. The UBE3A antisense essentially, in a schematic view, stops the production of the paternal copy of the UBE3A gene. So the gene gets started, but it does not actually fully transcribe or get produced in order to then make the UBE3A protein. So we all just make one copy of the UBE3A gene from the maternal copy, the maternal allele. And what happens in Angelman syndrome is that this maternal copy is either missing or mutated or not functioning properly. Now, this imprinting process, where the paternal copy is silent, only happens in neurons of the brain and spinal cord, which is why the rest of the body has a functioning paternal copy of the gene, but neurons do not have a functional paternal copy of the gene. And this is called a silent or imprinted gene, which is only imprinted in neurons. So what are the different gene types? What are the different types of Angelman syndrome that we might experience? So a neurotypical individual will have one copy from mom and one copy from dad, but the copy from dad is silent in neurons, like I just said. An individual with a deletion is missing an entire segment of that chromosome, which involves the UBE3A gene. So the copy from the father is still normal, but it is silent, like it is in all of us, but they don't have a functional copy from mom. An individual with a mutation, it looks somewhat normal, but the letters of the sequences are not normal. So there may be a substitution with a letter or a letter that is missing. And it ultimately results in the protein that the UBE3A gene normally makes to not be made at all. Or if it is made, it is not made properly. So it doesn't have the proper function. An individual with UPD or uniparental disomy, those individuals have two copies of the paternal allele. And so ultimately, both copies are silent, but both copies are perfectly healthy. But ultimately, there is no functional copy in the brain. In individuals with an imprinting center defect, that behaves very similarly to UPD in that there is a maternal copy and there is a paternal copy, and the gene itself is totally normal. The paternal copy is silent like all of us, but the maternal copy is also silenced because there is a mutation or a deletion in an imprinting center that makes the maternal copy behave like the paternal copy. And so 70 to 80% of our families are living with a deletion, where individuals, about 10% of individuals are living with a mutation, and about 3% of individuals are living with the uniparental disomy, and about 3% with an imprinting center defect. And the others that are diagnosed with Angelman syndrome typically are diagnosed clinically because they behave like Angelman, but the diagnosis genetically has not yet been proven. And so what is so important about the UBE3A gene? Well, all of you that live with individuals with Angelman syndrome know there is a lot of important things about the UBE3A gene. So the UBE3A protein that is made by the gene is essentially an enzyme, and that enzyme tags other proteins, and that is called ubiquitination. And it ubiquinates other proteins, especially within neurons and around neurons. It tags them in order to encourage them to be broken down or encourage them to be upregulated. So it essentially regulates the breakdown or function of other proteins that are around it. And so it is incredibly important. It's almost like the policeman of the space between two neurons, the synapse. And so that ubiquitination is necessary in order to balance or have homeostasis of the environment around neurons. And it's localized to the area of the neurons, the presynaptic area, which is, we have two, neurons speak to each other. So here's neuron number 1, which is called presynaptic, and neuron number 2, which is called postsynaptic. And it essentially regulates that space between the presynaptic and the postsynaptic compartments. A deficiency in UBE3A protein results in accumulation of unnecessary proteins and byproducts that are in this space, and the space between the two neurons is called the synapse. And if you have an abnormality in the synapse, that is called a synaptopathy. And ultimately, by having a deficiency in UBE3A, we have a synaptopathy. And the signals that are resulting in that synaptopathy is an accumulation of some excitatory signals and the failure to have inhibitory signals. And by not having inhibition, we get what we call a loss of tonic inhibition. And that's why our kids are super excited. They're excited emotionally, their brains are excited, which is why they have seizures. Their brains are excited, which is why they can't sleep. And they really can't understand it in the communication between the neurons. The neurons can't understand when to be excited and when to be inhibited. And so if we want to walk, we're going to activate one leg and inhibit the other leg. And imagine if that coordination is not possible because we don't have the ability to have inhibition. And so if we don't have the ability to have inhibition, imagine how difficult it is to talk. Because when we talk, we are activating hundreds and hundreds of muscles in our mouth, in our cheeks, in our in our throat, and we are inhibiting other muscles. And if we can't coordinate that activation and inhibition, talking is one of the most precise motor functions that requires activation and inhibition. So if you can only imagine how hard it is for our kids to be able to coordinate those movements. And speaking is one of the most intricate types of movements. And so by having this lack of protein, we have symptoms of Angelman syndrome. And those symptoms are relatively universal, but there is some heterogeneity or differences between individuals, especially individuals with different genotypes. But universally, unless you have mosaicism or some very unique mutation, individuals with Angelman syndrome universally have a lack of speech. They have life-threatening and debilitating seizures in most individuals. Some of them are more severe than others. All individuals with Angelman have severe developmental delays. They often have very poor motor skills and significant incoordination. They have apraxia and dyspraxia. So you ask them in the clinic to do something and they won't do it, and then the clinician turns around and they go ahead and do it. When they're demanded upon, it is very hard for their body to do what their brain is telling them. They have very unique behavior characteristics. Many of these individuals are so incredibly happy and sometimes hypersocial. Many kids can have sleep disturbances. There are some kids that sleep 2 or 3 hours a night for their entire life. And we know ultimately any individual with any genotype has the inability currently to live independently, which is one of the most devastating symptoms of Angelman syndrome. Many individuals have some feeding issues, especially early on in life, like right after birth and in infancy. And we know there's a significant clinical unmet need for this disease because there are absolutely no approved treatments for Angelman syndrome where we stand today. So first, I'd like to introduce you to FAST. The Foundation for Angelman Syndrome Therapeutics has a mission, and our main mission, our role, is to take parents and professionals that are dedicated to finding treatments and therapeutics for Angelman syndrome, and to create an aggressive research agenda, which is what we have done over the last 12 years. The Foundation's committed to assisting individuals living with Angelman syndrome to reach their full potential in life and to improve their quality of life, with the ultimate goal of bringing practical therapeutic treatment options into medical practices around the world as quickly and as safely as possible. And so FAST, up to date, has funded and raised over $20 million in spending in research and support services in order to push toward therapeutics for individuals living with Angelman syndrome. Over $3 million has been directed toward creating new animal models, like the large deletion rat, the deletion and mutation pig, a very large deletion mouse model, as well as different cell cultures and small mini brains like organoids. We have funded over 20 laboratories in order to help identify some of the etiologies behind Angelman syndrome and treatment options that can address those etiologies. We have helped to support over 6 gene therapy strategies or disease modifying strategies in order to help replace or activate the missing gene, as well as look at downstream targets in order to help improve the function of the different targets that are impacted by the UBE3 gene. If you look at this timeline, you can see that FAST was started in 2008. And here we are in 2020, where FAST helped to launch the first phase 1/2 clinical trial for a disease-modifying therapeutic, altering the genetic reading of the UBE3 gene. And so the goal of FAST has been to bring therapeutic strategies to human patients. And that is exactly what FAST has done over the last 12 years. And so we are very proud of that. And we have supported every other pharmaceutical company along the way. So what is our roadmap for success? Our roadmap has 3 main parts. The first is to investigate therapeutic strategies, whether that's through gene or protein replacement therapy or activating the paternal copy of the gene. So you can see here in the nucleus of the neuron, we are either replacing the missing maternal copy of the gene, or we want to activate the silent paternal copy of the gene. But we also want to identify downstream treatments. And so at the synapse, there's a lot going on that's dysfunctional, and that is because we don't have the ubiquitination of the UBE3 protein. So helping to ensure that we can find those downstream targets and see how they can benefit therapeutics for Angelman syndrome, as well as symptomatic relief, like anti-epileptic drugs and sleep supplements and neuroinflammatory inhibitors. Doing things that we can help the symptoms of Angelman syndrome is also incredibly important. Additionally, we are preparing for clinical trials. Sorry, let me just go back here. So we want to help prepare for clinical trials. By doing that, we created animal models, like I said earlier, the mouse and the rat and the pig. We helped to support and co-found the Angelman Syndrome Biomarker and Outcome Measure Consortium, developing endpoints and clinical measures that are appropriate for the Angelman syndrome population, of which very, very few exist until now. We want to help with clinical trial design for pharmaceutical companies. We know the patient population better than anybody because we're parents, and we can help design the clinical trials and help mitigate some of the risks of how those clinical trials can be executed. And we want to ensure that we do all of the testing we need to in every single genotype, so no child with Angelman syndrome is left behind. That we ensure one gene therapy strategy or one paternal activation strategy or one downstream target may benefit one genotype more than another. And we want to investigate all of them to ensure that every genotype is covered and is able to see the clinical benefit of potential therapeutics in the future. And so let's start with disease-modifying strategies. So gene replacement therapy is where we take, there's a viral vector, and typically this is known as an adeno-associated virus, an AAV, and we put a gene inside that AAV virus. The virus is a delivery mechanism that we inject into the spinal fluid with various different approaches potentially, and that needle goes into the spinal fluid. It injects this virus, but this virus is not pathogenic. So this virus is, all the bad parts of the virus have been taken out, and all the good parts that our kids are missing, like the UBE3A gene, has been implanted into it. The virus then takes the UBE3A gene, and it, what we call transfects, or gets into the neurons of the brain and spinal cord. And then inside the neuron, it can drop off the UBE3A gene. And it is created in such a way that the UBE3A gene can be made within that neuron, and it can be made so that it can ultimately make the UBE3A protein. And that's called gene replacement therapy. You can also do that in a way where you can deliver the protein. So you might be able to actually just deliver the enzyme into the neuron so that the enzyme itself can then perform the function of the UBE3A protein. You can also do that through a different type of virus called a lentivirus. Instead of that being injected directly into the brain, the lentivirus program is really delivered into the bone marrow. And so by taking a lentivirus and transfecting or putting the UBE3A gene into hematopoietic stem cells or young early cells of the bone marrow, we are able to then have those bone marrow cells making UBE3A. If those bone marrow cells are making a lot of UBE3A, those very easily cross the blood-brain barrier, and the bone marrow is a constant source of UBE3A production. As those cells, those hematopoietic stem cells cross the blood-brain barrier, they deliver the UBE3A gene into the space around the neurons. And there's something called cross-correction, where that secretory gene, which ultimately becomes a protein, can be taken up by the neurons and function inside of those neurons. And these programs have been really, funded. These strategies have been funded through FAST at UC Davis and University of Pennsylvania, as well as the University of South Florida, but they've now been taken up by various different pharmaceutical companies like PTC Therapeutics or Sarepta and many others, and we'll talk about that. So I just want you to be able to associate, when we talk about different programs and different academic institutions and different pharmaceutical companies who have a place in this landscape of Angelman syndrome research, you understand where we're talking about. So we're talking about gene replacement therapy. We're going to be talking about these different programs. The second option is paternal activation. And some people talk about stopping the stop. Okay, so we want to turn on the paternal copy of the gene and stop the stop. And we can do that through 4 main strategies that we're going to talk about. One is antisense oligonucleotides, like you just saw that gorgeous video. And we have 3 programs that are currently underway, 2 of which are in human clinical trials currently for antisense oligonucleotides. One is a program that is hopefully going to be underway in the next couple of years by Ionis and Biogen. One is in clinical trials right now by Genetics and Ultragenyx partnership. And one is in clinical trials right now by Roche and Genentech. So there's a lot of partnerships that are working to get this into kids as quickly as possible. Artificial transcription factors is a program that has been funded through University of California at Davis, and that is something that is active right now in the preclinical phase, as well as CRISPR technology, which we'll talk more about. And CRISPR technology has been investigated very largely through the University of North Carolina, as well as UC Davis, and now there's another program at the University of Pennsylvania. And AskBio has just taken on that program out of University of North Carolina. And then there's shRNA, or short hairpin RNA. And that's a way that you can take an adeno-associated virus and deliver a RNA so that you can inhibit the paternal antisense transcript and activate the paternal copy of the gene. And that program has been taken on by Taysha Gene Therapies, as well as Ovid Therapeutics. What about downstream targets? Downstream targets are also very important. And so Ovid Therapeutics has now completed a phase 2 clinical trial and is in the process of completing their phase 3 clinical trial. And this is a program that they'll talk to you more about today, which is essentially a GABA replacement therapy. It's a GABA agonist, and we'll talk more about that. Disruptive Nutrition has finished a clinical trial in the United States and is now moving into Australia and other countries with a ketone supplement in order to encourage ketone formation and ketone supplementation, which can decrease many of the symptoms of Angelman syndrome. NYU, as well as Neuron Pharmaceuticals, is supporting an IGF-1 and an IGF-2 program in order to address a synaptopathy, which is, we know, present in Angelman syndrome. And Neural Stem has a program with regenerative medicine looking at anti-inflammatory neuronal stem cells. So next, we want to prepare for clinical trials. And so how are we preparing for that? Well, one is that we're looking at the disease burden and the unmet need in Angelman syndrome. And so there's 2 great publications that came out, one by Ann Wheeler, and the other that just came out recently is the disease concept model that was put together through the Angelman Syndrome Biomarker and Outcome Measure Consortium in collaboration with Roche. And Tom Wilgoss did a really wonderful job putting this together, along with the support of parents and caretakers, where they were able to describe what matters to individuals with Angelman syndrome. And it was very clear that motor function, communication, behavior, and sleep were some of the main take-homes from that are really impacted by Angelman syndrome, and there is a very significant clinical unmet need with no current approved therapies. The NIH funded natural history study from 2006 to 2014 showed longitudinal data in over 300 individuals with Angelman syndrome, and then there's a new natural history study currently ongoing since 2019. The Angelman Syndrome Biomarker and Outcome Measure Consortium is a pre-competitive space that was co-founded by FAST that we brought in all of the pharmaceutical companies in the space, as well as all of the foundations. This was partnered with the Angelman Syndrome Foundation, as well as at the time Agilis Biotherapeutics, to really create this pre-competitive space. And we started off with 2 pharmaceutical companies in the space, and we now have over 15. So this has really grown quite tremendously to figure out what are some Angelman-specific endpoints that we can focus on. The Angelman Syndrome Global Registry that was founded out of Australia, out of FAST Australia, has really grown tremendously over the last 4 or 5 years, and that has really been hallmark into having the natural history data of Angelman syndrome by parents be updated on a yearly basis over time. And that has been created so that pharmaceutical companies can have access to that in order to be able to look at the natural history of the disease, to be able to compare, as they see their therapeutics moving forward, what changes are important and what changes are seen. And then finally, developing research models for Angelman syndrome of all genotypes. And that's through, like I said, animal models like the rat and the pig. We just funded Yale to create a large deletion mouse model where we can look at the entire UBE3A segment, as well as the 12 other genes surrounding UBE3A for 80% of our population that have a deletion. And then look at what the impact is of the other genes aside from UBE3A. So a mouse model that doesn't have UBE3A deleted, but has all the other genes deleted. And so this is an ongoing project that we're working with Yale on, as well as we have created a core infrastructure at University of California at Davis that has all of the rodent models, the rat and the mouse, as well as the iPSC lines. They'll have all of the organoid lines, and all of the genotype lines that are being produced by Yale will then go to the core research center so that pharmaceutical companies and academic clinicians can go to that center in order to test different drugs, different targets, and look at a robust type of research so that we can understand the benefits of these drugs and de-risk some of them before they potentially go into a clinical trial. So let's talk a little bit about the basis behind gene replacement therapy. So thousands of clinical trials are underway in humans using an AAV-based gene therapy. And so just some examples are Batten disease, Sanfilippo, AADC, spinal muscular atrophy. There's thousands, but not many of them are for neurologic disorders. And metachromic leukodystrophy and adenoleukodystrophy are some of the lentivirus-based gene therapies that have been looked at and have been published in human clinical trials. So we have really been able to repeat phenotypic rescue over and over again using gene therapy and gene replacement therapies. And that has been through the University of Pennsylvania Gene Therapy Program, which is an AAV program, as well as the University of California Davis Gene Therapy Program, which is a lentivirus program. And of course, out of the University of South Florida, Ed Weaver, who was really the first person in the space at all to publish on gene replacement therapy that could have a potentially huge meaningful impact in Angelman syndrome, where he was able to rescue cognitive benefit in the mouse model. The delivery is through viral vectors, like I said, and those viral vectors are able to transfect neurons of the brain and spinal cord, and that is injected through the spinal fluid. We have other types of programs that we are currently looking at. There are over 4 gene therapy-based programs, and these are just some of the examples of that in Angelman syndrome. And so some of the research, this is the first hallmark paper that came out of Dr. Weber's lab, looking at the mouse and giving an AAV delivered gene therapy. And really what he was able to show, and this is back in 2011, was that by increasing levels of UBE3A in the brain, you were able to improve some of the cognitive deficits in an adult model, a mouse model, with Angelman syndrome. And that was really, really promising to show. You can see here, the UBE3A is the brown, and you can see here, this is Angelman syndrome with no UBE3A. And you can see this is neurotypical, this is wild type. And all you need to know is that brown is really good. And if you can see that, you can't even tell the difference between this Angelman mouse brain and this Angelman mouse brain segment. You can see that UBE3A is being produced. And that is incredibly important. And you can see in this graph as well, we were able to normalize the protein levels to wild type levels. And so these are some functional tests where this is a water maze where you were able to see here that normally the mouse will cross over this water maze, they'll cross onto this platform and find the platform so they can get out of the water. Normally in Angelman syndrome, they don't really learn to go onto that platform, and they just keep exploring the water and never get to rescue on that platform very quickly. After treatment, you could see that the Angelman syndrome mouse was able to find that platform after they were able to learn how to, where it was. They were able to then circle that platform much more than was possible in the non-gene therapy treated animal. What about gene replacement therapy through stem cell delivery? Well, in 2013, a beautiful paper was published on metachromic leukodystrophy. And this was published in a disease that is neurodegenerative. And in this neurodegenerative disorder, if you look here at these individuals, you can see that this is before gene therapy in a 16-month-old child. This is a degenerative disease. And you can see the degeneration that happened from 16 months of age before gene therapy to an individual that did not receive gene therapy at 39 months of age. And you can see the black is bad, the gray is good. So you can see the degeneration that occurred at 39 months without gene therapy. All of this black is essentially holes in the brain that was degenerated. Well, look at the individual at 39 months that received a gene therapy. The gray here is so much more tremendous than the gray here. And that shows that this gene therapy was able to get into the brain from a bone marrow infusion of a lentiviral delivered hematopoietic stem cell project, and it was able to rescue the degeneration that was happening over that period of time, which is just incredible. And so we funded Joe Anderson's lab at the University of California, Davis, to look at this type of program where he was able to transfect the hematopoietic stem cells with a modified UBE3A gene using a lentivirus. And then what they do is they basically transfect these hematopoietic stem cells, which belong in the bone marrow. They give a dose of chemotherapy so that they can wipe out the bone marrow and then replace the bone marrow with your own stem cells. that are populated with UBE3A. So it's not like a standard bone marrow transplant because you're not receiving a transplant from another donor. You are getting back your own cells, but you are depopulating the bone marrow cells without UBE3A and repopulating them with UBE3A. The reason is that those cells cross the blood-brain barrier. When they cross the blood-brain barrier, now we can create this thing called cross-correction, where the neurons of the brain can take up that UBE3A that they didn't have before, and now they can receive UBE3A for the rest of their life. And Dr. Anderson will present to you at this meeting, but ultimately what he was able to show, and he presented this last year, was full phenotypic rescue in the mouse model of Angelman syndrome after this treatment in both newborn and full adult, equally able to rescue rotarod, digigate, beam walking, the EEG for seizures, and open field, and novel object recognition. And this is just one example that I find to be incredibly promising, is if you look at the Angelman syndrome mouse, you can see that they really just can't keep up on the digigate or on this little treadmill. If you look at it after stem cell treatment with hematopoietic stem cell transfer, you can see here that they are able to walk much better and they are able to keep up with the treadmill, which is incredible. Enzyme replacement therapy is essentially doing the same thing, but giving the enzyme that the kids are missing and giving it directly into the brain. And then they can hopefully have that functional protein. We are funding UC Davis, and this program is in the preclinical stages, but underway. And then paternal activation. When we look at paternal activation, like topoisomerase inhibitors, we have different programs that have been investigated, and it all started in 2011 by Dr. Ben Philpott out of the University of North Carolina. That moved to 2015 when Dr. Art Beaudet published a paper in Nature looking at antisense oligonucleotides for the treatment of Angelman syndrome. And since 2012, Dr. Scott Dindo has been working on a similar approach. Artificial transcription factors can activate the paternal copy as well, out of UC Davis and Dave Siegel's lab. And then more recently, shRNA programs have been started out of Taysha Gene Therapies and Ovid Therapeutics. Dr. Ryan Butler is the PI working on that program. So paternal activation, we have paternal activation of the allele, like I said, and this was the paper that came out in 2015 from Art Beaudet's lab, showing that we don't have any UBE3A activation in an Angelman syndrome mouse model here. And with an antisense oligonucleotide where we activate the paternal copy of the gene, you can see red is good. We are fully activating throughout the brain UBE3A, which is really fantastic. And we're also able to show that in the brain, this effect with a single high dose in the mouse brain was able to last 16 weeks or 4 months and then take down. So a single injection could potentially last a very, very long time. He was able to show that he rescued some of the cognitive deficits in the adult mouse model, and this was accomplished through a high dose of an ICV or an intrabrain injection in the mouse. And in humans, the focus is to do that, but through a lumbar puncture or a lower spine puncture like that video showed. And there are 2 clinical trials underway that started in 2020, the genetics trial and the Roche trial, and hopefully Ionis is well on their way as well. This can also be done through zinc fingers, and this is, again, this red shows that there's not as much UBE3A, and then after artificial transcription factors or zinc fingers, the red is much higher, showing that you can activate the paternal copy of the gene. The nice thing about this is it's non-invasive. It's a subcutaneous injection that crosses the blood-brain barrier, but there are some optimizing that needs to be done for a program like this because it is not as specific as something like an antisense oligonucleotide or CRISPR technology. CRISPR, which we hear a lot about in the news, is clustered regularly interspaced short palindromic repeats. So repeat that 7 times without a lisp and see how far you can go. But ultimately, this is a paper that just came out of Mark Zilka's lab by Dr. Wolter, and this came out just recently in the last month out of Nature. And really what they were able to show here was that by targeting a specific sequence, just like an antisense oligonucleotide with a guide RNA using an AAV vector, you are able to rescue phenotype in the Angelman syndrome mouse model, and you're able to activate the paternal copy of the gene. And so you can see here the UBE3 levels, and this is wild type, this is normal. This is Angelman syndrome, which is very, very low. And you could see after the CRISPR technology, they were able to activate this in the cortex by about 40%. You were able to get that, get that protein expressed, and you could see in the hippocampus as well. And so you're getting some protein expression in the animal model, and that's all it took in order to have very nice phenotypic rescue in these animals. And so they found that after 2 injections early in life, they were able to rescue phenotype for over 17 months where the paternal activation was still present. And so that was really promising. University of California, Davis has another program using Cas13, and the University of Pennsylvania has another program using Cas9. What about downstream targets? Well, we already talked about OV101 and gaboxidol. Ava is going to be speaking a lot more about this, about increasing GABA at the junction in order to improve functions where we can improve that tonic inhibition, like sleep and learning and memory and motor function. So there's a lot of promise, and this was published in 2012. The ketone supplements, which was published in 2016, increasing ketone bodies in the brain can decrease seizures and improve learning and memory. And then just recently in 2020, Dr. Alberini's lab published looking at IGF-2 ligands by correcting a synaptopathy, and she had amazing phenotypic rescue in her mouse model of Angelman syndrome. And so the primary goal is that each therapeutic target can potentially benefit people in different ways. And different individuals with different genotypes of different ages may benefit by different approaches or different combinations of approaches. And so the ultimate is to investigate each option very thoroughly. And we don't stop until we have a symptomatic cure or meaningful translational therapeutic success. And so we start with proof of concept, we move to safety assessments, then to clinical trials, and then hopefully successful treatments that are transformative for our patient population. And so what matters to caregivers in order to do that? Well, the disease concept model, like I already talked about, but we also did a survey in January of 2018, and FAST did this survey, and we questioned 332 parents responded to this survey. And what they found was that the most important change that would be meaningful for an effective therapeutic for their child would be improvement in communication and speech, number one, by far, the number one response. Second to that was seizures, and third to that was mobility, improved motor function, for both gross motor and fine motor. And so that really helps to drive the goals of the Angelman Syndrome Biomarker and Outcome Measure Consortium, in which we're going to be looking at different focus areas like EEG, cognition, communication, development, and we're going to try and come up with endpoints that are Angelman syndrome specific. And so that's how we help to bring therapeutics to humans. And so we think about how we're bringing therapeutics to humans. We are looking at these, a map that looks a bit like this. So we start with basic research. We have a proof of concept. We have a preclinical plan. And then from there, we are thinking about how the FDA is going to look at that for a human candidate. We're going to bring it to the FDA, and that's going to be through IND enabling studies. Once we bring those studies to the FDA, then the FDA needs to approve a clinical trial starting. That is a huge landmark. When they approve a clinical trial starting, then we're going to start either in a phase 1 clinical trial, which is just safety, or a phase 2 clinical trial, which is looking at safety and early efficacy. Many of those are called phase 1/2 clinical trials. A phase 3 is a much larger trial looking at safety and efficacy. And then finally, FDA approval for a drug. And so 5 years ago, when I joined this community, this is a little bit of what the map looked like. And so 5 years ago, we had one program looking at gene therapy, and that was Agilis. We had one ASO program that was still in basic research, and that was Ionis. We had an artificial transcription factor program that was still in an academic institution, University of California, Davis. And we had some downstream targets that were being investigated, like what Ovid Therapeutics has put through, as well as disruptive nutrition for a ketone supplement. And so that's the landscape 5 years ago. And like I said, this is really the landmark space of doing IND enabling studies and getting through the FDA to start a clinical trial. Let's fast forward 3 years ago. So 3 years ago, we went from this to this. Another program came in. A stem cell project started. The ASO program moved, and another ASO program started. The artificial transcription factor project moved, and a CRISPR program started. The downstream targets moved, and we had 3 more people in the space. And this is our space there. So now we have 2 programs that are in that space, 1 in a human clinical trial. What about 2 years ago? Well, that gene therapy program moved, that gene therapy program moved, another one got into the space. The stem cell program moved fiercely, the ASO programs moved, and another one got into the space. Artificial transcription factors moved, CRISPR moved, another one got into the space. The gene therapy program, the downstream targets moved, and another one got into the space. So this is very, very exciting. And now we have 1, 2, 3, 4, 5 programs in that space. What about 1 year ago? AAV moved, another one came into the space, and another one came into the space. Stem cells moved, ASOs moved, and moved. ATFs moved, CRISPR, more CRISPR, another CRISPR program. Our downstream targets in phase 3 clinical trial, finishing up nearly a phase 2, and another one's moving forward. And then another one came into the space, and here we are. Where are we today? Okay, today, AAVs are moving, but they're not moving quite as fast because gene therapy takes a lot more time in this space, this preclinical space for patient safety, for human safety. Our stem cell program is moving. ASO programs are moving. One started a clinical trial, a second started a clinical trial. ATFs are moving, CRISPR is moving, one program moved, another one moved. Downstream targets, an shRNA program started. A second shRNA program started. Our downstream target program with Ovid Therapeutics is approaching the end of their phase 3. Ketone esters are moving into other countries, and other downstream target programs are moving toward IND. And so if you look at this map now, compared to where we were 5 years ago, it is breathtaking that we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 programs in IND-enabling studies or in clinical trials. 4 are in clinical trials, and a 5th about to start. Is just breathtaking for where we were 5 years ago. But this takes time, and we have moved this so beautifully thanks to the community. So bringing therapeutics to clinical trials requires safety testing in large animal models. We need to really ensure we're doing it right, because usually these infusions are one and done. Turning on the paternal allele, we need to find the ideal point to inhibit. We need to ensure that we test this in larger animal models to know what's maximal benefit, and we want to minimize toxicity. We need to find the candidates that are the most long-acting, that have the highest knockdown and the most gene activation. Downstream targets, we want to ensure we're looking at the best outcomes so that they can be approved by the FDA if we're seeing meaningful change. And we need to be clinical trial ready by getting these endpoints ready. We need to continue in the drug discovery place, because every day technology is changing, and we need to keep up with that technology in order to explore all new technologies and all technologies for all genotypes. So what can you do now? You can participate. The Global Angelman Syndrome Registry is a great way for you to participate. The natural history study is a great way for you to participate. A new natural history study is just up and running now in the UK. A great way for people overseas to participate. The stem cell project at Yale, all it requires is a blood draw. If you want to participate, contact the folks at Yale or contact us and we can get you in touch with them. Participating in endpoint development, like the communication measure, the ORCA, the CGIAS that's under development, EEG studies by Biogen and Roche, the CSF study by Biogen and Roche, participating in the Freesia study, which is the endpoint study, and talking to your doctors about participation in trials is most important. Talk about what you want. So we're investigating therapeutic strategies in our roadmap. We are identifying downstream targets, and we are preparing for clinical trials, because therapeutics are surrounding our community, and AS is clearly the most promising disorder right now to treat. We have more people interested in Angelman syndrome now than ever before, and I would argue maybe more interested in Angelman syndrome than any other neurodevelopmental disorder right now. We need to welcome all players to this amazing community, because our community is nothing short of amazing. But what's more important for me is that each of you parents understand that knowledge is power. You need to understand everything for your child, because if you understand, you can ask questions. And if you can ask questions, you know your options. And if you know your options, you are going to make an educated decision for your kid. And that is the most important thing for me, is that you listen to facts. Understand and ask questions, and don't be shy. That's what I am here for. That's what we are here for. Participate when you can and where you can. Talk to your doctors. But most importantly, we need to celebrate. We need to celebrate the hope for the future of all children living with Angelman syndrome. There are so many diseases and disorders that will never be cured or treated in our lifetime. But with all of our help and all of our support and this entire community, I am confident Angelman syndrome will not be one of them. Thank you for your time.